We are going to continue from where we stopped the last time. We talked about um, structures and functions of cell organelles. So far we've talked about the cell, we've described the animal and plant cell. So, in this video, we are going to continue from the structure and function of the cell organelles. So we have the first organelle we're going to be considering is the cell plasma membrane. Is the cell plasma membrane, and you see in the plant cell, actually, this cell plasma is actually preceded by the cell wall. It's known as a cell wall. So, and it's a this rigid structure that you can see out here is this rigid structure that you can see outside of this place here. So we have the cell wall. So I said this region right here that you're seeing here, that is the cell wall, this part that encloses the inner part of the cell. Whilst this point here, these portions here, are the portion we call the cell membrane. The cell membrane, this boundary here, this one, that is what we call the cell membrane. All right. So now we're going to be looking at the cell membrane, the function of the cell membrane first. Now, the cell membrane is the outer covering of a cell. That is for the animal cell. Now, if it's the animal cell, it will be the outermost covering. But if it's the plant cell, it's going to be um, the second layer of the covering of a cell. In the plants, you are going to find the cell membrane in the second layer, while in the animal cell is the first layer, right? That separates the cell from the external environment and neighboring cells. So the cell membrane essentially the separates cells from one another. So it separates the inner environment from the outside environment and from other cells, right? So and it controls and regulates the movement of substances in and out of the cell. It also supports and protects cells from damage and leakage. So one important thing I want to notice here is that it says it regulates the movement of substances in and out of the cell. So it is semi-permeable. Is that okay? It is semi-permeable. And one thing about this is that, about the cell membrane is that it is a living layer as in contrast with this cell wall, which is a dead layer. So this cell membrane is a living layer, while the cell wall is a dead layer. And so that is why this cell membrane, or called the plasma membrane, that is another name, the plasma membrane, actually is used for, it has components on it, some other macromolecules that are used in in communication with other organelles of the cell. So that is why it is separated from other cells. So it has its own unique. It has its own. It is by itself living, but it functions together with other cells by way of different signaling and macromolecules, signaling systems, macromolecules that are attached to its external surface. Now, on its external surface, as you can see in this diagram, you're going to be seeing these sugar chains that is uses for communication with other cells. These various sugar chains, they are called sugar side chains, and they are used for both signaling and communication with other cells. All right, and then you're going to see this head and tail region. So this head region is called hydro the hydrophilic. The head is hydrophilic. It means it is this is hydro. Hydro means the word hydro means water, and the word philic The word philic is got from the word filio, which means love or likeness. So hydrophilic means water loving. So this head is a water loving head, and this tail is hydrophobic. So phobe or phobos is the word hate, the Greek word hate. So hydrophobic means water hating. So these tails hate water, and this head love water, and that is why, because our internal environment, not our internal environment is actually made up of what, of water, chiefly water. So other molecules are suspended in those waters, as well as the cell. So the external environment of the cell containing water um, will, will actually require the cell to have a a kind of surface that can interact, that can that can interact, that can actually survive in that surface. So both internal and external environment of the cell are composed of water. So internally the cell, so this is the cell. Outside, which is the outside of the cell, is composed of water. Um, and here is also composed of water. So our polar, basically polar um, solutions, solutions. So this guy here will have to be such, membrane as can interact with this internal and external environment. So that is why you see this external surface. Let me clean it a little. This external surface is going to be hydrophilic, and this surface here also is going to be hydrophilic, so that water in the internal. So if this portion here is the inside of the cell, this environment here is the inside of the cell, it will not be affected. The internal environment here, this hydrophobic tail here will not be affected. All right, so that is all we have for the cell membrane. It has both hydrophobic heads and hydrophilic heads, or polar heads. Is that okay? That's another word. Polar head. So polar region to interact with um, the polar liquid that is outside of the cell, all right, and inside of the cell as well. All right. So we can also see cholesterol, which is a type of fat and oil, of fat and oil, or lipid, um, you know, um, you know, um, which are embedded on the surface of those phospholipid bilayers. Um, so so that is, so we call them phospholipid bilayer. I forgot to mention that this hydrophilic end. So the lipid is the lipid layer is, I said, phospholipid. So the lipid layer is this hydrophobic end. I've talked about this in a video where I talked about the um, peroxidation of lipids. So I talked about the hydrophobicity of lipids. So lipids are hydrophobic, and phosphate group are hydrophilic. So this phosphate group are attached at the ends, and these ones that are in blue colors here are the the um, the fats, and the yellow colored ones are the cholesterols. Cholesterols are also a special type of lipid. Is that okay? So then they also have embedded in them the integral proteins. So you see, so those proteins are actually attached to, I mean, the sugar side chains that you see, they are attached to the proteins. So we call them proteoglycans. Proteoglycans. All right. So proteoglycans, proteo for the word protein, and glycan for the word glucose, or referring to carbohydrates in general. So proteoglycans are embedded in between in between the cell membrane. So if I have the cell membrane this way, so in between, just for communication and cell signaling, as I talked about, cell signaling. All right. So these guys are attached to the side chains. Those are sugar side chains are attached to the proteins. All right. So that will be let's proceed. Let's proceed to the next one, the next slide, which is the cell wall. The cell wall. The cell wall is the most prominent part present only. So take note of the word only. So present only in the plant cell. So it is only the plant cells I mentioned earlier that the have the cell wall. So I said the cell wall. This is the boundary here. This boundary. So and I mentioned the fact that it's a dead layer. It's a dead layer, and it is freely permeable. Freely permeable. All right, but let's talk about the 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 composition. All right, number one thing, the function is the fact that it protects. So its major function is the protection and support. Protection and support. It stands as a rigid structure for mechanical support of the plant cell. So that's why you naturally see that plant cells or the plants are usually rigid throughout. Plants is rigid throughout. So it's because of the presence of a cell wall cell wall that protects the plasma membrane and other cellular components. So the plasma membrane, which is the cell membrane, is protected by this layer. So we, in the animal cells, I earlier said the cell membrane comes first, but in the plant cell, the cell wall comes first. Now it is made up of cellulose, hemicellulose, and pectin. And this is very important for you to note. It is made up of cellulose, hemicellulose, and pectin. Right? All of these are sugars. All of these are sugars. Um, this is, a very long molecule of sugar. The cellulose and the hemicellulose, they are very long molecules of sugars that are in fact indigestible by um, by that are only digestible by ruminant animals. All right. So and the cellulose and the hemicellulose are what form the hard structure, the the hard the rigidity that gives this cell wall its rigidity. Then it provides shape and support to the cell and protects them from mechanical shocks and injuries. So we've talked about that as a major function of the cell wall. All right, let's proceed to the next, which is the next layer that will come across when we proceed into the cell. So we've left the outermost part, which is the cell wall. Then we went into the cell membrane, which enclosed the cell. And now we are into the cell cytoplasm, which is we call it the cytoplasmic matrix. That is the fluid in which all organelles are suspended. So it is the cell, it is the cytoplasm that contains this fluid in which the other organelles of the cell are suspended. All right, so it is a thick, clear, jelly-like substance that is present inside the cell membrane. And it is the site where chemical reactions within the cell takes place. So all chemical reactions take place within the cell cytoplasm. And the cell organelles are suspended here, as I said. So this blue matrix you're seeing here is the cell membrane. This blue portion here is the cell membrane. All right. And these organelles are floating around and around. These organelles are floating around in the cytoplasm. All right. So that is it for the cytoplasm. The next one is the nucleus. All right. So this is one which is the most important organelle of the cell, the nucleus. All right. In fact, the word nucleus means central. All right. Central or core. Is that okay? So that is in the. If I say it is core, it means it is very important. Means this, and that's why it's centrally located because it controls almost, not almost, in fact, it controls all activities of the cell, controls all activities of the cell. So let's talk about what it contains. It contains the hereditary materials of the cell, which is the DNA. So it contains the DNA, and it also contains a special um, structure known as the nucleus, which is specially stained. It is stained differently from other parts of the cell. Now, it is enclosed within an envelope. It's called the nuclear envelope. The nuclear envelope encloses the cell membrane. So, remember we said the eukaryotic cells, eukaryotic cells carry on means enclosure. So, the eukaryotic cells possess membrane-bound organelles. So, nucleus is a membrane-bound organelle. It is bound by the nuclear envelope or nuclear membrane. That's another word for it. Nuclear envelope or the nuclear membrane that separates the nucleus from the rest of the cell. All right. It protects the DNA and it is an integral component of the eukaryotic cell. And its main function is that it controls all activities of the cell. It is called the brain of the cell. The brain of the cell, right? So that is the nucleus. So, and it has, you can see, let's uncover the the structures. So you can see here, there is a nuclear pore, so it means that there are holes in between it. And now this is why there are holes, is because in the this is the nucleus. I say it's a specially stained organelle. So you see, this nucleus. Um, if you check, probably going to link that video in the description where I talked about cell division and all of that. So I mentioned some of these things and cell replication and duplication, cell replication and transcription, DNA replication rather, and transcription, where I talked about how that the RNA, the ribosome is being synthesized by the RNA, and then RNA synthesizes protein. So I talked about the fact that nucleus is the site for the production of what? Of ribosomes. So these ribosomes then go out through these pores. So the ribosomes go out through they exit the nucleus via the pores. We call them nuclear pores. Nuclear. So whenever you hear the word nuclear, it means it's referring to the nucleus or the center of something. Is that okay? So nuclear pore. Then we have the chromatin materials. Chromatin materials are just packaged DNAs. Packaged DNAs. That is DNA structures that have been um, um, organized separately, right? And you see, this structure is called the nucleoplasm. Nucleoplasm, right? It is the structure that you know that gives the entire nucleus its own structure separately. So it's called the nucleoplasm. All right. So let's view the other organelles. Um, We also have the nucleus, which I have talked about, and it is a site for ribosome synthesis. So it synthesizes or produces ribosomes, and is involved in controlling cellular activities and cellular reproduction. So nucleus function majorly for ribosome synthesis. So for your JAM, and most of the time in your post-UTME, you are going to be asked, you are going to be the question is going to always be about if it's for function of nucleus, it's always ribosome synthesis, even though sometimes um, it plays a role in cellular reproduction. It is majorly the site for ribosomal ribosome production. All right. Then we move to the next one, which is the ribosome itself. So what is the function of this ribosome that is produced in the nucleus? Now, the function of the ribosome is that it is where protein is being synthesized. Okay, so it does mean that I'll have to link that video where I talked about these things again, ribosome synthesis, from replication, transcription, and translation and transcription of DNA. Ahead, transcription and translation. Transcription, oh God, translation and transcription of DNA, right? So, where protein is being produced, protein is being produced, and in the ribosome, via we talked about the messenger RNA bringing the message from the nucleus rather, from the nucleus, then the transfer RNA begin to copy the codon sequence of this messenger RNA into protein, into different amino acid chains, right? So that is just it in a jiffy. I'm going to link that video in the description, right? It is found in both prokaryotes and eukaryotes, right? So in prokaryotes, in both prokaryotes and eukaryotes, you're going to see it attached to, in prokaryotes, you're going to see it scattered. So it is scattered around the nucleus, around the cell in prokaryotes, but in eukaryotes, it is also scattered around the cell, but it is organized on the endoplasmic reticulum. It is organized on the endoplasmic reticulum, so that one is called the rough ER, and also you're going to see it just around the nuclear membrane, because of course it escapes, comes out through the nuclear pore, so you have to find around the nuclear membrane, right? Then we also have, okay, it's also the prokaryotic ribosomes are much smaller than eukaryotic ribosomes, all right? Yeah, because of course the eukaryotic cell is much more larger than the prokaryotic cell, um, as we discussed earlier. So definitely the ribosomes are, in fact, most of the cell structures of the eukaryotic cell are relatively larger than the prokaryotic cells. So, so it is expected that even the ribosome should be greater um, than those of the prokaryotic cell. All right. We have the next organelle, which is the Golgi apparatus. Now, the Golgi apparatus is an organelle that was named after a man called Golgi, Camilo Golgi. Camilo, I hope I spelled it well. Camilo Golgi. So he was the one who discovered the Golgi apparatus, or Golgi apparatus, Golgi actually, Golgi body or Golgi apparatus. So it forms what is called a Golgi complex. So this entire structure is called the Golgi complex. And just like the endoplasmic reticulum, this guy does engages in the function of processing substances in two directions, which we call the trans and the cis direction. Trans, the word trans means different. Is that okay? Different, or from one place to another. Is that okay? Changing her, like from one place to another, or different, something like that. Is that okay? Then the cis means same, cis, same. All right. So if it processes stuff it can process substances in trans phase. That is, if the stuff enters through this place and go out through the other direction, or if it enters through or in the cis phase, so it enters and goes out through the same direction. So that is just what that is talking about, the trans and cis phase of the processing of substances that the Golgi apparatus or Golgi complex altogether perform. So the cell's post office, as it is, okay, we call it the cell's post office. That's one thing, that's one very important term. Just like we call nucleus the cell brain of the cell, mitochondria powerhouse, we call these guys cell's post office, as is involved in transmission of materials within the cell. Right. In fact, these guys also process stuff outside the cell, not just within the cell. Endoplasmic reticulums are the ones that are restricted to within the cell, transport transportation of materials. So, and it is important to note that there is a theory about how that these guys are degenerates of endoplasmic reticulum, but we'll not be getting that here in this video. We'll not be getting into that detail, and that's why you see that their structures are essentially alike. Their structures are alike. So there's a theory behind the fact that it is old endoplasmic reticulum or some endoplasmic reticulum that are change their function, they become Golgi apparatus. All right, but that is another thing entirely. Just know that they are called the cell post office, and they are involved in the formation of lysosomes. All right, so the word lyse meaning split. Okay, I guess we're going to get into it in further sections of the functions of the cell organelle. So it forms the lysosome essentially. So you can see this vesicle that is pinching off here is going to essentially form a lysosome. I mean, most of the time it forms lysosome. So these are also secretory vesicles. So vesicles always pinch off of this Golgi apparatus, and they do so in either the cis direction or the trans direction. All right. Then helps in collecting, packaging, and distributing molecules synthesized at one location in the cell and moved to a new location in the same cell. Stores and secretes glycoproteins, right? So they store and secrete glycoproteins, right? So those are some of these things that they will process into secretory vesicles that you find there. All right. We have the next one, which is the endoplasmic reticulum. The endoplasmic reticulum is responsible for transportation and metabolism of substances. So they metabolize carbohydrate, proteins, and steroids throughout the cell. So they are restricted within the cell. I've talked about that. So notice this diagram here. This one is called the rough endoplasmic reticulum. It is rough because of the presence of ribosomes. It is rough because of the presence of ribosomes. I repeat, it is rough because of the presence of ribosomes attached to them. So, and that is why some textbooks might tell you that rough endoplasmic reticulum also function in the production of proteins. Actually, they are not the one producing the proteins. It is the function of the ribosome to produce those proteins. But because those ribosomes are attached to them and they already function in the cell logistics, all right, logistics. So already they will therefore be able to transport those proteins that are produced by the ribosomes. All right, I hope that is clear. They don't produce proteins by themselves. The ribosomes do, but the ribosomes, since they are attached to the endoplasmic reticulum, they transport the proteins that are produced in the ribosomes throughout the cell. Now, another thing to take note of is the fact that you see there is a very close, there is this closeness between the ribosome. So notice this endoplasmic reticulum and the cell and the nucleus rather. You see that they are very close, aren't they? So look at them closely. You're going to notice that they are very close. So, and that's why it is when this guy produces the ribosome comes out through the nuclear pore, and the nuclear pore and the ribosome attaches itself first to the endoplasmic reticulum before even getting out into the cytoplasm of the cell. So the rough ER is associated with the nucleus and carries the ribosome, while smooth ER exists freely in the cytoplasm. So also the theory I was talking to you about is such that we have the rough ER. The rough ER, after losing its ribosome, will become the smooth ER, and then the smooth ER, after growing for a while or growing to a particular stage. So sometimes it's not just that they have grown to a particular stage. It is just the way they form and become the Golgi apparatus. So from here, this guy becomes the Golgi apparatus. So this becomes this. That's just the way the theory explains it. All right. So, and they also synthesize lipid, steroids, and protein. So, okay, so we just mentioned that they synthesize protein, but they what they do to the protein is only modifications in a way. So, but the major production of protein happen in the ribosome. All right. Now, the next one is the mitochondria. All right, so this is my big guy here. I like him a lot because he's a powerhouse of the cell. All right, and what does it do? It produces the major energy currency, which is the ATP. The energy currency of the cell is the ATP, and it's produced in the mitochondria. So you see, in fact, this mitochondria is a very big guy, and I say that because you see there is a theory that is also known as the endosymbiotic theory. Endosymbiotic, endosymbiotic. All right. So you can browse about that. I don't know if I'll be dealing with that anytime soon, but it is just the theory that proposes that these guys, membrane-bound organelles such as this guy, mitochondria, were existing alone. They were existing as independent cells until um, you know um, they needed until they needed some they needed protection, things they needed things like protection, basically protection. So they needed to attach um, to prokaryotic cells, in order to now form the eukaryotic cell. Right. Well, you can browse more on that in the symbiotic theory. Today we'll just be dealing with what mitochondria does. So you can see this rough structure. This rough structure is called the systeme, that is the cristae rather. And the structure of the endoplasmic, okay, let me point that out. The endoplasmic reticulum, that structure that looks like this is called the cisternate. Cisternate. Cisterna. Cisterna for singular. So cisternate for plural. So while that of the mitochondria is called the cristae. These infoldings, these internal infoldings, we call them we call it for this guy, we call it the cristae, while for endoplasmic reticulum is called the cisternate. Cisternate or cisterna for one, singular. All right, then there is an inner membrane and there is an outer membrane. It also has its own fluid, so notice it's like its own cytoplasm, and that was what the theory of endosymbiosis, that was what brought about the theory of endosymbiosis. It's like these guys can actually exist on their own. So it has its own matrix, we call it the mitochondrial matrix, which is more like the cytoplasm of the cell. In fact, it has its own DNA, call it the mitochondrial DNA, call it the mitochondrial DNA. And inner foldings called cristae is to increase the surface area. So this infolding increase the surface area of the inner portion of this guy for more ATP synthesis. All right. Then enzymes responsible for fatty acid oxidation are located in the matrix of the mitochondria. So fatty acid oxidation, the oxidation of fatty acid, I talked about. I'm going to link that video in the description also. Where I talked about oxidation of fatty acids. The enzymes that are responsible for that function, oxidizing fatty acids, are called are located in this matrix. All right. I mean, okay, they are actually called fatty acids. So those are lipids. So they'll be called lipases. No, the ones that break down, the ones that break down, um, that break down fatty acids are called lipases. Okay, so but the ones that oxidize fatty acids. I'm going to be before the end of this video. If I do remember, I'm going to mention it. I'm going to mention. I think I've forgotten right now. the enzymes that um, you know oxidize that oxidize these guys fatty acid. And if not, I probably mentioned it in that video. So I'm going to link that video in the description below. All right. The next one is the lysosomes. Okay. So I did talk about it. The lysosome. All right. So the word lys means to destroy or to split. To destroy, and soma means body. So it means they are bodies that split apart or destroy. So the way they in fact, they have their own special name. We call them suicide bags. So it's, okay, yeah, they are mentioned there. Okay, that is mentioned there. The cells suicide bag. So they are surrounded by nuclear envelope that separates the nucleus from the rest of the cell. No, no. This is a mistake. This is a mistake. Excuse me. Um, these guys, lysosomes, are actually, you know, you know they have they have a membrane also, so which is a single, okay, they have a single wall membrane, and that's the function. They have enzymes inside them that enables them to break down substances. So they break down substances. After breaking down those substances, what they do is they either break those substances down to to um to sizes, I mean to chemicals. I say sizes, I'm sorry. Chemicals that are useful to the cell. So chemicals that will become useful to the cell, or sometimes if they cannot be broken down into chemicals that are useful, then they what are transported out. They're transported outside of the cell. So, and lysosomes are, you see, the major question that usually come out of lysosomes are the organelles that possess them. So they are most abundant in cells like the cells of the liver and sperm cells. So those very active cells will possess, um, oh God, the cells of the liver, yes, and the cells of the sperm, yes. They're going to possess lysosome, and also the I forgot to mention something. I think I remember something about the ribosome. Now is that I'm sorry for digressing. The ribosome in the human, the ribosome in our cell, in the animal cell, is about 40%. In the human cell, now when I remember fully, I'm going to tell you. I'm going to let us know. There's just something I remembered about lysosome now, that about ribosomes now, that is one of the possible questions that could be asked in both JAMB and post-UTM. They are involved in secretion of digestive enzymes. So they aid in digestion. In digestion, you break down food substances. So um, they aid in digestion. All right. So the next one is the chloroplast. So one important thing is that it is found only in plants and algae. So algae referring to blue-green algae, and of course the red algae and red algae rhodophyceae, and um, uh, the brown algae, that is phaeophyceae. Right, I'm going to link that video also in the description where I talked about them. All right, then the fluid within this chloroplast is called the stroma. So this blue portion here is called the stroma. You see, these are called thylakoids, singly thylakoids. Then this arrangement is called a granum, and many of these are called grana, as plural of granum. And then the thylakoid, these thylakoids are called thylakoid discs, and then there is this guy that connects them. We call it the lamella. This guy that is um, connecting these guys, connecting each stack of We call them stack of thylakoids. Stacks of thylakoids. So the stack of thylakoids is called the granum. So this lamella connects each granum. Now there's an inner membrane and there's an outer membrane. Then this space here is called intermembrane space. There's an intermembrane space just around it. Yes, this is the intermembrane space. All right, and make sure you're focusing on the video, please. The fluid within this, okay, yeah, I mentioned that each coin-like structure is called thylakoid and stack of thylakoid is called, okay, I mentioned that. And the next one is the vacuoles, all right? So very important to note that the vacuole is present in both plants and animal bodies, most conspicuous in plants, all right? Very big and centrally located. So in fact, the nucleus of the plant cell is actually shifted to its side because of the size of the vacuole. Please notice that it is shifted to one side, the nucleus, but because of the large size of the vacuole. And the vacuole is enclosed within what is called a tonoplast, that is the covering the outer membrane the of the vacuole. So, and the vacuole is just fluid filled. It's just filled with is a fluid. Okay, this is a fluid sac bound by a membrane known as the tonoplast. Okay, it is much larger and it contains sap. It's called the cell sap. Cell sap. All right, cell sap is basically contained of saline solution, um, um, some 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 other it's composed of carbohydrates and proteins and saline water, saline fluid, right? So those are the compositions of the cell vacuole. All right, the next one is the central, which is going to be the final one I'll be discussing. Okay, actually, I'm going to discuss one more thing, but I didn't add it to the slide. I just remembered now. The central is the one that plays a major role in the cell division. All right, now look at the function, the structure here. I want you to notice something about the structure here. This structure here is the way it arranges itself, is the way it places itself in the centrosome. So, and this arrangement is very important. The two the two centrioles are positioned 90 degrees to each other, directly perpendicular to each other. So, and that is for it to be able to perform its function of arranging spindle fiber spindles. We call them. They are not spindle fibers. They are actually we call them microfilaments. Is that okay? Microfilaments, and that's what I want to talk about again. Microfilaments are organized by this centriole. It is on the microfilaments that the the chromosomes will come to attach themselves to via their kinetochores. You see, I'm going to link the video of the cell division here in the description below. Please make sure you check out on those videos so that you're going to understand fully all of these things I'm talking about. They work during mitosis and meiosis, right? And they are found primarily in plant cells and some lower plants, animal cells and some lower plants. They are usually present near the nucleus, but only visible when the cell is dividing, right? So they are closer to the nucleus, as you can see here, but you can only see them conspicuously when the cell is dividing because they are one of the only remaining organelle. In fact, if I did not the only remaining organelle when the cell is dividing, they are the only remaining organelle. When the nuclear membrane disappears. So another thing is the cytoskeleton. The cytoskeleton is just made up of um, um, actin, microfilaments, and microtubules. They have the cytoskeleton. That's the other thing I want to talk about. Is made up of actin, microfilament, and microtubules. microtubules. So you see, these microtubules are smaller than actin fibers, while actin fibers, no, the microfilament is the largest. I'm going to look at, okay, the major thing is this, that both of these help in giving the cell its shape. So even though they are usually found at the boundary of the cell, of the cytoplasm, close to the cell membrane, inside of the cell. So, and the function is in keeping the